Mr. Vidasak, half of biotechnology research and development office, Department of Agriculture, Thailand. Okay.
Okay.
Today, Mr. Vidasak will talk in the topic of development of the in-house genetically modified with 17180 reference plasmid for qualitative detection by Japex real-time PCR. Okay, Mr. Vidasak, please.
Thank you very much for introducing myself. And as you know, the topic today is about the GMO implant. So first, I would like to introduce my working place. I work for Department of Agriculture Thailand, Biotechnology Research and Development Office. We consist of 4 groups. The first group is agriculture, biotechnology. Second group is gene banks. And the third group is mushroom R&D. And the last group is GMO detection. I work for GMO detection here. And now we have 2 main offices. The first office in Pathum Thani province. Also, the gene bank is here. And the second one is in Bangkok, the GMO laboratories. I'm working here in Bangkok. It's located in Kasetsart, University Bang Khen campus. Thailand, we have Plant Quarantine Act start in 1952, and we have a lot of amendment version. The last version is in 2008. The purpose of Plant Quarantine Act is to protect environment, protect human health, protect native species, and protect genetic resources. We have notification system in case of unexpected GMOs bring to Thailand. We allow GMO import to Thailand only for research purpose. If someone needs to bring GMO into Thailand Kingdom, have to follow the DOA guideline, which is comply to Cartagena Protocol on biosafety. Fortunately, GM corn and GM soybean are permitted to import to Thailand only for raw material industry use only. But another species is prohibited. So now you can see the picture, left-hand side, the biosafety guideline of the NTDS, and the right-hand side, the green color show the biosafety guideline of the DOA. And you can download in the orange one from internet, and the green one you can come to my office. I can give you the book of biosafety guideline. And now very hot issue of genome editing and in plant, the regulation is now under consideration process. Here show the mission of the GMO laboratory of the DOA. First, we have R&D for GM detection technique, and we have the service work on screening service for 16 samples per year, and we improve laboratory under ISO IEC 17025. We improve the service under the certified survey from customer. We have surveillance for GMO contamination in Thailand. We go through the country and collect the sample randomly and detect if it's GMO or not GMO. And also the last one, we have a safety working group for the DOA. And also we are under the ISO standard laboratories, and also we are authorized lab for the private lab. For example, Central Lab Thailand, ASL, Omics, SGC, and also Intertech. Under the ISO standard, we have 4 scopes. We have corn and its product, soybean and its product, rice and its product, and papaya and its product. And now we are planning to expand our scope to wheat and its product. So many research. So now my research will support this ISO standard to expand the scope in wheat. This is show the process in our lab under the ISO standard. First, come from the sample submission and the sample legislation is in the system. We have sample preparation, DNA extraction, DNA concentration, and quality check. And the last one, we have target gene detection. And after get the result, we have the issue test report. And the finally customer can receive the test report. And also we have guideline for sampling of the GMO sample in the left-hand side here. Here shows sample of our test report. After customer get the test report in left-hand side here, they can request for the certification of non-GMO in the right-hand side here, and they can use this certificate as the attached document for exportation or importation plan product to Thailand. Here shows some example picture of our lab here, the sample in almost the plant production sample, and also so the sample preparation, DNA extraction, and gene detection. Our lab, we use the life cycle 480 load system for the real-time PCR. And also you can see this machine is so powerful, it can detect in different wavelengths, like 4 or 5 colors in the same time. So why wheat? Why I'm doing research on wheat? Because we import a lot of wheat every year. For example, in 2019, we import wheat to Thailand more than 3,600 million kilograms, is like billion Thai baht. And also we use wheat in many industries, in food supply chain, for example, in wheat flour, instant noodle, and bread, in bakery, and also cracker, and also some in animal feed. So how about GM wheat? How it come? So I would like to start from Google, and then I typing the ISAAA. So the result you can find ISA.org is the International Service for Acquisition of Biotech Application. It's a non-profit international organization that share agricultural biotechnology focusing on the genetic engineering. Then I click. The web page shows the GM approval database. Then I click the GM approval database. Here show a lot of species in the species of plant that record to have GM approval all around the world. And then I click the last one, wheat here. After click to wheat, you can find only one event of GM wheat found is MON 71800. Then I click here. After click, you can find a lot of information of this wheat is produced from Monsanto and is at glyphosate herbicide tolerant with CP4 EPSPS gene insertion. So I'd like to have the brief history of wheat GM. It started in 90s. Glyphosate tolerant wheat developed by Monsanto, and MON 71800 was authorized to have the field trial in USA. And after finishing the program, no presence of Monsanto 71800 in commercial market. But in 2013, unexpected, MON 71800 was discovered and noticed and confirmed in Oregon by the USDA. So after that, European Joint Research Center, EUJRC, released the test method and the method validation for MON 71800 qualitative detection. And this means the European country have really worried about this GM wheat explode. Unfortunately, MON 71800 CRM certified reference material is not available in commercial market. So I decided to make in-house reference material of MON 71800. I designed the fragment of insertion consists of CAMV35S parameter, ACC1, is the specific endogenous of wheat, and MON 71800 event specific element, and NOS terminator, and also ampicillin resistant gene, and the restriction recognized region between each gene. Then I cloning this fragment to PUC 57 vector and transfer to E. coli competent cell and extract plasmid and use it for the reference materials. This slide I show the sequence of insertion fragment. The red color show the position of primer and probe of each gene, and the green color show the position of restriction enzyme recognized region. Here I show the sequence of the primer and probe which used in this research. You can see in different 4 genes have different probe label colors. In CAMV35S parameter, I use FAM with the BHQ1 quencher. In NOS thermometer, I use hex with the BHQ1 quencher. In MON 71800, I use Texalate with the BHQ2 quencher. And ACC1 gene, I use cyanide-5 with the BHQ3 quencher. This means each gene can be detected in different wavelengths. This slide shows the reagent used in PCR reaction. You can see in Simplex real-time PCR use same concentration of primer and probe, but in Tetraplex real-time PCR reaction, the concentration of primer and probe of each gene need to be adjusted. And after optimization, the final appropriate concentration of primer and probe shown in the table here. DNA extraction is also one of important steps. Here I show the result of DNA extraction of different kind of sample, consists of wheat flour, instant noodle, and cracker as well. The results show good amount of and quality of DNA. Also, we did the inhibition test of the extracted DNA. The results show no inhibitor found in DNA by the delta C less than 0.5 and R square is higher than 98%. This shows the comparison between Simplex real-time PCR and Tetraplex real-time PCR in different concentration of reference material of each gene. The crossing points of Simplex real-time PCR and Tetraplex real-time PCR show significantly different. For qualitative detection, the result is enough to interpret result because when we report the result, we report in the detection or not detection. This table shows the limitation of detection of the PUC 57 MON 71800. So I compare 1 picogram and 0.1 picogram of reference materials. In 0.1 picogram show detection result less than 90%. So I make conclusion that the LOD of the PUC 57 MON 71800 is 1 picogram in the Tetraplex real-time PCR reaction. Here I show the advantage of multiplex real-time PCR. The Simplex have detect endogenous gene by the PCR and do the gel electrophoresis and detect CAMV 35 parameter one time, detect NOS thermometer one time. But in multiplex real-time PCR, you can detect all 4 genes in just one reaction. It's faster and cheaper than Simplex real-time PCR. Here I show the comparison of working flow of the Simplex and multiplex real-time PCR. In multiplex working flow, we can cut off the endogenous detection step out, and we can save time of the gene detection from 4 days to 1 day. And here show the benefit of the multiplex real-time PCR in our lab. So we can reduce the cost to 32%, and it's safer and less contamination from pipetting, and reduce the working time in the detection to 60%, and reduce the time for all process is 42%. And also under the ISO standard, we have to do the customer certified survey every year, so we can keep 5 score from 5. So I want to make the conclusion here in how GMV MON 71800 reference.
Hello.
Yes.
อาจารย์ขออนุญาตที่ไม่ได้เป็นแชร์คือไม่มีที่มีวิลากรมีสัญญาไม่มี
Okay, here is my conclusion. Thank you. Maybe the time is running out.
Education on the room. a question? Okay.
Excuse me, I have one question.
Okay. Please.
Hello, can you hear me?
Yeah.
Yeah, I hear.
Okay, I think I see in the result in table 4, you show the CT value of the simplex versus the tetraplex. Yes. And this table. And I'm not sure the t-test will show the difference significantly, right?
Yeah.
I think the simplex and tetraplex should be not different, should be similar.
Yeah.
Why the simplex and tetraplex show the difference significantly?
Yeah, because the reaction is different. Simplex have just one pair of probe and primer, but tetraplex have 4 pair of probe and primers. So this is actually the preliminary experiment results. So now we are adjusting to the condition, but for a qualitative result, we can report if the CP is higher than lower than LOD, we can report to be the detect of the GMO.
Oh, okay.
Yeah.
You will report only the qualitative report, right?
Yeah, qualitative only.
Okay. Thank you for your answer.
Thank you.